Hi everybody. Today we're gonna have our third lecture for third class, and today we're gonna talk about the macromolecules and chemical composition of cells. So let's begin. Primarily we're gonna talk about the proteins, fats, carbohydrates, different types of carbohydrates, and we're gonna talk about their structures, and I'm gonna explain different types of experiments and how can we find out what types of molecule we have: protein, carbohydrates, and this is also those. I'm gonna show how to do those experiments in a virtual lab and try to explain the results we're gonna obtain, because this is one of the most important labs we're gonna have. Um, the final and midterm is gonna have a lot of questions based on this lab. So guys, please take this seriously. Some of the information you have to learn and know from that lab. All right, let's begin. So most of the molecules, so all of the molecules made from atoms, and those atoms make micromolecules. So we all made from different types of molecules of like a building blocks. Um, For example, proteins have a different building blocks than carbohydrates, like sugars. One of the most important molecule I will say we have um, five, about 4 to 5 very important molecules. Not molecules, actually, elements, atoms, right? The first one is going to be carbon. So this is a carbon. The first one of the most important element is a carbon. The second is hydrogen, nitrogen, and oxygen. So this is four elements. One of the most important, and they basically, if you're gonna take just the hydrogen, carbon, nitrogen, and oxygen, they're gonna make up probably 90 to 99, more than 95% of your body composition. So all the molecules in the body, you can imagine, they're gonna have nitrogen, carbon, hydrogen, and oxygen. So those are the building blocks of our body. But the carbon, one of the most important. This element, any molecule which is have a carbon is gonna called organic compounds, right? So most of the tissues we have, most of the molecules we have are organic in our body, and they have carbon. So we're gonna talk about the carbohydrates, lipids, proteins. We're not gonna talk about the nucleic acids. So carbohydrates gonna be divided in monosaccharides, polysaccharides. We're gonna talk about this. I'm gonna explain things about lipids, and proteins are gonna divide by amino acids, polypeptides, and just peptides. So first we're gonna talk about the protein. I'm gonna explain what is proteins. So first of all, proteins is one of the most important molecules. This is a molecules, right? Those molecules made from elements, element made molecules, and proteins are one of the most important component of your body. So if you're gonna see the ratio of your body composition, the protein is gonna make up one, like the biggest majority of your body composition. For example, your muscles. The muscles is the big chunk of your body. So this is just an illustration how the portion of a protein looks like. I'm sorry about this. So this is a portion of a protein, and each the protein is made up of a building blocks, and we call them amino acid. So each separate amino acid is a single building block to make a protein. And we have 20 different amino acids. You don't have to know those structure, right? This is a molecules, and those molecules, when they interact, right? When they each different amino acid is an interact, they're gonna make a chain, and this chain is a portion of a protein. So you don't have to know those, guys, if you take in biochemistry or cell biology, molecular biology. Probably you're gonna know them, and definitely if you're gonna take biochem, you have to know them, structure, names, and all, and the rest of for now, just remember we have 20 different amino acids, and the structure to make those so basically we have 20 different amino acids which are required to make any proteins, right? Some of them we have essential and non-essential amino acids. So they I don't remember right now exactly how much. It's like, I think it's a ratio like 11:9. It's a 9 essential and 11 non-essential, but I might be mistaken. But the point is, we have essential amino acids, which is your body is required to make other different proteins, and non-essential amino acids. So non-essential amino acids you have to obtain from your diet and from your food, and non-essential amino acids can be produced and synthesized by your body. So to be able to make different types of protein by your body, your diet has to be well balanced. So all the different types of proteins have to be consumed with your food, so the body will be able to produce different types of proteins, because as I said, the majority of chunk of your body weight is made of by protein. Most of the cells, as we already talked about the structure of cells, all those organelles are made by made of proteins. So the cells is the mass majority of a cell, or a single cell, is a proteins. So the well-known source of a proteins, it's a meat, right? Fish, and some vegetables have some amount of protein, but they're not as rich as, for example, meat and fish. But things like nuts, they have a lot of protein. So vegetarian people most of the time consume a lot of veggies, which is high on the proteins, to consume those non essential amino acids to be able to actually live. All right. So let me talk about. So this is a new concept I'm gonna introduce. So please know this. This is gonna come up. 1 or 2 questions is definitely gonna come up on a final on midterm. So, and this is dehydration in hydrolysis reaction. So this is basically, this is one of the most simplest reaction in chemistry we have. And what's the purpose of this reaction? So when we have 2 molecules, so for example, one amino acids and the other amino acids, right? And we're gonna make a chain of those 2 amino acids to produce a portion of a protein. When you're gonna take one amino acid and connect with the other amino acids, you're gonna make a peptide bond. So this is a peptide bond between carbon of first amino acid and a nitrogen of second amino acid. And this bond is called a peptide bond. Please remember this. So the bond between two amino acids called peptide bond. Remember this. And when you go uh, from the left to the right, this is called dehydration reaction. So what is a dehydration? From this vocabulary, you can actually translate what does that mean. Dehydration, low water, right? You don't have water. So what it means, you lose water. So in this reaction, you lose water. So where the water come from? The carbon has oxygen and a hydroxy group, which has oxygen and hydrogen. And your nitrogen has two hydrogens. So the molecular formula of water is H2O. You have two hydrogens and you have water. This is a molecular formula of water, which is right here. So this is where you lose, this is why it's called dehydration, because you lose water when you have one molecule plus second molecule. Those hydrogen is going to bind to OH and is going to make water. So this is a dehydration reaction. And the hydrolysis is actually the opposite reaction. When you're gonna break that bond, one molecule water is gonna come, right? You're gonna break the peptide bond between carbon and nitrogen. One molecule water is gonna come. It's a plus, right? So hydrolysis, it's a plus water, and it's gonna be divided and end up in those places, right? So please remember this reaction: dehydration, we lose water; hydrolysis, dehydration, we lose water, and we actually produce more complex molecules. And the hydrolysis, when you kind of destroy the chain of some kind of poly chain for singular building blocks. So hydrolysis, you use water and you break the chain for building blocks. In dehydration, in the the opposite. So this is protein. We have like 50 or like 100 amino acids. So let me introduce that as a vocabulary. So we have protein and we have a peptide. So as you remember, the peptide, we have a peptide bond, remember? So the peptide bond between two amino acids. So you have a protein and a peptide. So but now this is peptide is different than peptide bond, right? So we have peptide bond and just a peptide and protein. So make sure you know the difference between three of them. So the protein, it's a sequences of it's a sequences of chain of amino acids which has more than 50 amino acids, more than 50 building blocks. If your chain have more than 50 amino acids, it's called a protein. If you have less than a 50, it's called a peptide, right? And then a peptide bond, this is where you have interactions between two different amino acids, right? Peptide bond, then you have a peptide. This is less than 50 amino acids. And then you have a polypeptide or protein. Polypeptide or protein is have more than 50 amino acids. And the way we're going to test this, we're going to have a burette reagent. And when the solution, you have a solution, right? You add the burette reagent, and it's gonna if the solution has a protein, it's gonna turn purple. If solution is gonna have a peptide, it's gonna turn pink. Or if you have a high concentration of a peptide, if you have a really high concentration of a peptide, it's also gonna turn purple. All right. So please remember this one more time. The protein, if you have a sample and you test with a burette reagent, the burette reagent by itself is blue, and if you have a protein in your sample, it's gonna turn purple. if you have peptide instead of protein, if you have a peptide and you test with a bioreagent, it's going to turn pink. But if you have a high concentration of a peptide, it's going to also turn purple. So this, I'm just going to explain the different types of a structures of a proteins, right? So we have a primary structure. The primary structure is just a sequences of amino acids, chain of amino acids, right? And then those and then you have a secondary structure. So the secondary structure is alpha helixes, beta sheets, and it's also beta turns and random coiled conformation. This is 4, but we have only 2 of them. You only know you just have to understand the idea of what is a protein, right? So the protein is very complex, extremely complex structure. And as you imagine, if you have 20 different amino acids, that sequences could be, you can come up with any sequences you want with a protein, and it is limits for size of a protein, but basically you can come up with any structure with any function. That's why we have tremendous amount of different types of protein in our body. Most of the proteins in our body have been identified, they have been sequences, but it is still some proteins in our bodies, because it's so many of them, we still don't know their structures, we still don't know their primary sequences, right? And then you have a tertiary structure when different types of secondary structure made a 3D structure. So the tertiary structure is a functional 3D structure is a functional structure of a protein. And if you have more than one, if you have very complex proteins, sometimes you might have a subunits of a tertiary structures, and they're gonna make a quaternary structure. So the quaternary structure is more complex protein, and tertiary structure is a little bit more simple proteins. So the tertiary structure proteins, they have a very specific 3D shape, and this very specific 3D shape has specific function. So the function come from the shape and different types of amino acid. So if you're gonna destroy this 3D structure, and this 3D structure gonna come back to the secondary structure or primary structure, your protein is not gonna be functional no more, right? So only functional structure of a protein is a tertiary and quaternary structure. If for some reasons you have a quaternary structure and you're gonna go to the tertiary structure, this protein is not gonna be functional, because it has to has all those subunits together, right? If you have a protein that has a tertiary structure, and for some reasons you have a high temperature, very acid environment or base environment, it's gonna denature, right? And if the protein is gonna denature, it's gonna go to the secondary structure, primary structure, and it's gonna lose its function. So there's a term you have to know, denaturing. So when the proteins is gonna denature, lose its native structure. What is denature, right? When it's protein, it's gonna lose its native structure. It's gonna go to secondary structure and primary structure, and it's gonna lose its function. So please remember this. And how you can do this? You can apply very high heat if you want to boil some eggs, or if you want to boil meat, right? Or sometimes if you're gonna if some of you cook and know how to cook meat, if you're gonna put lemon juice on the meat for example overnight, what's gonna happen with the meat? The meat is gonna be more it's gonna lose its tendency. So the meat now is gonna be less um, how to explain this. So when you're gonna apply the acid, from the lemon or different types of juice, it could be tomato juice. The tomato juice and lemon juice, they has very low pH, and this pH is acid. So what's gonna happen? Those the proteins on that meat, they're gonna lose their tertiary structure. And when they're gonna lose the tertiary structure, first of all, they're gonna break down, right? You're gonna actually break down the protein. The nature is also, if you're gonna break down proteins, it's also, you may refer them as a denaturing. So this is the same. Lose native structure, breakdown, denaturing is synonyms in this example. So you're actually gonna break down the proteins, and that's why those meat, after you apply the lemon juice, is not going to be stiff. It's going to be you're going to be able to chew it very easily. So the next one, we're going to talk about the carbohydrates. So carbohydrates. So as I said before, the proteins made. Oh, before we're gonna go, I want to introduce as a concept of proteins. A lot of proteins have very different type of function in our body, right? Some of the, for example, proteins like hemoglobin, which is in your blood, is responsible for transportation of oxygen. So different proteins have very specific function, like transportation, or motility, which is help your muscle to move, right? Some of the proteins actually can contract, and that's why you can walk, and so on. So some of the proteins are enzymes, which is can speed up different types of reaction. Some proteins are actually can break down the other proteins, right? So this is we have a tremendous amount of different proteins, and most of them have very specific function, and the majority of your body made by proteins. So the next one is a carbohydrates. We're going to talk about the carbohydrates. We know we have simple and complex carbohydrates, but what does that mean? So the simple carbohydrates, such as fructose, glucose, and you can find those simple carbohydrates in a simple sugars like fruits, cakes, and all the processed food, unfortunately. The more complex polysaccharide, such as starch, cellulose, come from grain and plants, and most of the people do not like eat them, which is very bad for you, and we'll explain later why. So, and why do we need carbohydrates? The main reason we're gonna talk about the carbohydrates, this is a molecules which is we use to fuel our body, right? We use proteins as a building blocks. So if you want to gain some muscle, right, if you want to get bulky, you have to eat a lot of carbohydrates and a lot of meat. But if you just want to live your life and you want to stay in your weight, you just have to consume a normal amount of complex carbohydrates just to fuel your body, right? But if you want to gain some muscle, you have to eat a lot of building blocks, such as amino acids, which you can find in a chicken, fish, right? To help to build your proteins. And to be able to build your proteins, you have to have an energy, and this energy comes from the carbohydrates, simple and complex. So so the difference between simple and and complex carbohydrates comes from their names. So when you have a simple carbohydrates, it means their structure is very simple, and when you have a complex, it means the structure is more complex than from simple. So let me just go and explain this. So this is one molecule of a glucose. We have two molecules of glucose, and this one glucose is an example of a simple uh, carbo simple sugar, right? So this is a simple. So one more time. Fructose and glucose are simple sugars. Where you can find the glucose you can find in no, let me go, let me explain from that way. So glucose, it's a simple sugar, right? The structure you have as 6 carbons. One, actually it comes from that one. 1, 2, 3, 4, 5, 6. You have 6 carbons, and those, so it's C6H12O6. This is a formula of glucose. I don't ask you to remember this, but be familiar with this formula. This is like universal formula, and if you go into medical field, you have to know this. The same as water and alcohol. You should know formulas like that. So if you want to build up, right, this is exactly the same principle as in the proteins, exactly the same, right? You want to take two simple building blocks, in this case it's a 2 glucose, and you want to make a more complex structure. And right, so you're taking 1 glucose plus 2nd glucose, you're gonna lose water. This is called dehydration, right? You're gonna lose water and you're gonna make a bond, glycolytic bond. This is called glycolytic bond. You don't have to remember this. So now you have a monosaccharide plus monosaccharide. So what is mono? Mono, it means one. Saccharide, it means sugar from Latin, right? So what does that mean? You have one sugar plus one sugar. You have disaccharide. What is disaccharide? D, it means two. Saccharide means sugar. You have two sugars. One sugar plus two. 1 plus 1, you have one complex structure, which is half. This is, they not separate, they now it's a one structure. You have to understand this is one molecule right now. This is 2 molecules, this is 1 molecule, and it's called a disaccharide. So it is still simple sugar. So we consider a complex sugars if it has more than just two, if it has more than 2 simple sugars, right? If you will have third glucose, it will be already complex sugar. But if it's 1 or 2, so monosaccharide and disaccharides are simple sugars. If you have a complex sugar, it has to have more than 2, 3, 4, 5, and so on. And dehydration reaction exactly works exactly the same as in a protein. What comes, this glycolytic bond is going to break down and is going to gain the hydrogen and the hydroxyl group to their own place. So dehydration reaction is a chemical reaction in which something is joined together by the formation of covalent bond, and what is produced during the reaction, used to connect monomers together to make polymers, right? So this is monomer, and this is monomer, and this is demonomer, or if you will have the third one, it will be polymer. So please know these different vocabularies. As an example, formation of starch, which is a polymer which is have more than 3 glucose sit together, right? And the hydrolysis is a reaction where it's a chemical reaction in which water molecules are added to break a covalent bond. So please know this, go back and watch this and try to understand and remember this. So this is an example of a starch. So starch made by glucose. So this is a pure glucose which is stuck together to make a polymer. Polymer of a glucose are called starch, right? So this is it's very big molecule. Even though it's one, it has many glucose, but it's a one molecule because all of them are covalently attached to each other. And where you can find the starch, starch you can find in most of the vegetables, and some of the vegetables very high in starch. For example, potato, sweet potato, carrots, pumpkin. Those loaded by starch, and the starch is very helpful for you. And let me now introduce the I really want to talk about this, about the why simple and the difference between simple and complex carbohydrates, and why it is important to consume complex carbohydrates. So first of all, let me introduce and repeat again. Simple carbohydrates I include simple sugars as a glucose is just one monomer, and a fructose. So where you can find those two in a table sugar. So the table sugar is right here. It's actually disaccharide, which is made by fructose in a glucose. And also fruits. You can find fructose in the fruits, and it also has glucose too. This is the probably corn syrup, which is loaded by fructose and glucose. But what happens with the body when you consume those molecules? Simple molecules, right? So first of all, when you're going to consume those simple molecules, because they're very simple, right? They small. If you're going to compare these molecules, from just one glucose, the site is completely different. So what do you think is going to happen? What do you think is going to be digested first? Just one glucose, or one glucose molecule, or the huge simple chain? The answer is obvious. The one glucose is going to be absorbed by your body really quickly. And when simple sugars as a glucose and fructose is going to absorb by your body very quickly, it's going to spike your insulin. The insulin is going to go to your blood, it's going to open up the cells, and those carbohydrates, especially glucose, is going to go into your cells. And I have to mention, your brain is one of the most energy needed portion of your body. So the brain alone consumes 25 to 35% of all your energy in the form of glucose, right? So if you're just gonna sleep and you just watch Netflix and do nothing, right? Your brain alone is gonna consume 25-30% of all your energy in the form of glucose from your food and your diet. But then we can't. So, and we know the high spikes. But when you consume a lot of sugars, the high spikes of insulin is very dangerous. So those actually they're gonna block. Uh, so when you always have high spikes, first of all, your cells is gonna lose sensitivity to your insulin, and you might develop diabetes, which is extremely common nowadays. And they also say this disease of rich countries, because if you're gonna go to places where poor people cannot afford sugars, they don't have a diabetes. So this is a problem of rich countries and people who doesn't know anything about diet. So glucose is actually good for you because glucose is going to be absorbed by all your cells. Your muscles is working on glucose too when you run, and your brain is one of the main um, organ which is consume glucose. And remember, the table sugar, 50% fructose and 50% glucose. But what happened with the fructose? Actually, fructose is gonna go to your liver, and it's gonna be consumed only by liver, and because all the other cells in your body cannot take uh, fructose because it doesn't have a special protein to um, bring that molecule to inside the cell. So actually your muscles cannot use fructose, your brain cannot use fructose. But what happens if you're gonna consume a hundred grams of a hundred grams of a table sugar, for example, right? This is maybe like 2 liters of Coke. This is how much sugar you have, maybe even more. So the 50 grams of a glucose, right? Remember table sugar, 50-50, 50 of glucose, 50 of fructose. The 50% of glucose is going to consumed by your cells in your body, but very high. It's going to be all those 50 grams is going to go to your bloodstream. They're going to spike insulin. This is very bad for your body. So this is high spikes of insulin is very bad. But what happens with the rest 50 grams of a fructose? This is actually your liver is gonna take the battle with that. So the fructose is gonna try to digest, but the way the liver is process fructose, and it's only way it does, is gonna turn those fructose to the fat, and those fructose is gonna go and stay. It's gonna uh, transfer, and it's going to change to the fat. The molecule of fructose is going to change the fat, and that's why it is very, very unhealthy to use any types of processed sugars, uh, because the half of processed sugars contain high amount of a fructose, which is going to be absorbed really quickly, extremely quickly, because it doesn't have anything, right? It's just a sugar. It's going to consume by your bloodstream. The liver is going to take a huge chunk, and it's all it can do is turn to the fat. But the other side, if you have fruits, if you're going to consume fruits, the fruits have fibers. The fruits have fibers in it, right? And uh, vitamins, so it has a lot of benefits. So when you're going to consume fruits, those fructose and glucose, first of all, is not going to be absorbed by your body as quickly as is just table sugar. Those fiber is going to slow down this consumption, and those when you have those fructose, when this fructose is gonna absorb by your liver, and it's gonna process slowly to turn into the fats, and some of the fat might be used by your body and and be used by different types of cells. So when you're gonna consume fruits, it's a different story. So it's not gonna be as bad as just consuming simple sugars in a processed food. But in the complex sugars, we have long chains of a glucose, right? And as we said already, glucose is more healthy than the fructose because glucose can absorb by every cells in your body, but fructose can absorb by liver and only. So if you're gonna consume things like pasta, potatoes, sweet potatoes, grains, those guys are made by complex sugars in a form of starch. And those starch, when you have an oatmeal in the morning, right? Those your enzymes is gonna chop those glucose molecules at some time, right? So your body is gonna consume. For example, you're gonna have 100 grams of oatmeal. and 100 grams of oatmeal is probably gonna have like 70 grams of a glucose, but that glucose is not gonna get absorbed right now, like at the same time, because you have to, first of all, you have to chop those molecules, right? And the glucose is gonna absorb very slowly at very steady level, steady rate, right? It's gonna absorb by steady rate, and your insulin is not gonna get spiked. So you're gonna have increase of insulin, but it's not gonna get huge if all those um, glucose will be separately and they will be absorbed at once. But all of them can be, because you have to remember the glucose by itself is it's a small molecule, but the chain of a glucose is very big, so it cannot be absorbed by your body. So you have to chop them off, right? You have to break down this molecule to simple molecules, and then it can be absorbed. That's why when you absorb complex sugars, they're very healthy, because they introduce glucose at at a slow rate, at steady rate, and it's gonna spike your insulin. Uh, the way we store glucose and carbohydrates in our body in the form of a glycogen. So glycogen has very similar structure as a starch, but we don't. The human body store the starch in two places: is your muscle and your liver, and total amount is vary between 300-400 grams per body per person. And then these and approximately is gonna enough to fuel the body like 24, like 30 hours, right? After 24 hours, your glycogen is gonna be depleted, and you're gonna use and you're gonna get really hungry, and you have to consume some food to fuel your brain, because your brain works on glucose, which is come from the starch and simple and a glucose. You can find glucose from different places, but the brain runs on a glucose. You cannot feed your brain with the proteins on the fats. It doesn't like this. It's not gonna work in well. If you're gonna have just a meat-based diet, you're gonna go crazy, because your brain is not gonna work on this stuff. That's why people who have a diabetes, sometimes they have seizures, because uh, the body produce a lot of keto bodies from when they use fat and protein as a fuel, and the brain doesn't like this. So that's why sometimes you have seizures, because when you have diabetes, you cannot use um, sugar from your bloodstream, because your cells stop being sensitive to sugars, and your cells cannot uptake that sugars, that glucose, and it has to choose proteins and fat for the fuel. But when it does this, it's very toxic. And the next one is starch. I'm sorry, it's cellulose fibers, right? The cellulose, we cannot digest cellulose. You have to have a special. So if you're gonna see those bonds, look up, and in a starch, in a glycogen, it goes down. So the difference between those bonds makes us unable to digest the cellulose. And what is cellulose? Cellulose you can find in a wood cell wall. The plant cell wall have a made of cellulose. That's why it's very tough. You have to the way the animals like cows and horses can be able to eat grass and be able to digest and receive glucose from that. It's actually one glucose, because they have a special bacteria in their gut, and those bacteria produce proteins and enzymes, which is going to help them to break down those bonds and release glucose to be consumed by the body. The humans do not have those bacteria. That's why we cannot consume grass and live on that. So the carbohydrates: glucose 1 sugar unit monosaccharide, maltose 2 sugar units disaccharide. This is still simple carbohydrates. And then you have a starch. It's a polysaccharide. Starch, glycogen, and cellulose. Those are the three examples, and this is a polysaccharide. This is a complex carbohydrate. The way we're gonna test this is we're gonna use Benedict reagent, and when you're gonna add the reagent, the more and this is test for more mono and disaccharides, for simple sugars, right? This is test for simple sugars, and the more you have simple sugars, the more red it's going to turn after the heating. When you add the Benedict reagent to your substance, the more you have, the more red color is going to be, from green to red, but after the heating. And then test for polysaccharides is you're going to use iodine. It's going to go from brown to bluish black or like very dark color. So the next one is the fats. Let me introduce the two vocabulary. This one, I think everybody heard the healthy fats and healthy fats, but what does it mean? And it comes from saturation of those bonds. So those bonds can be saturated and non-saturated. So if you have a saturated bonds, it means you have just one covalent bond between those carbons, and this is just an example of a fat, right? Fat molecule. And unsaturated fats, they have a double bonds. And when you have unsaturated bonds, those tails are gonna have a kink, right? So so you have some space between those tails of a fatty acids chain, right? You have a space, and if you remember, our membrane is made from fat. Phospholipids fat. What it means that we have a phosphate polar hat. We have a backbone of glycerol. This is a backbone of glycerol, and we have two fatty acids chain, right? Two fatty acid chain. And so the reason why we have polar hat and we have a polar hat and non-polar hat. So the polar hat can interact with water from inside and outside, right? And then non-polar chain gonna non-polar chain tails gonna interact with non-polar chain. So we have bilayer of a fat of a phospholipids. So we have two layers of a phospholipids in a cell membrane, one from outside and from the inside, but they turn their heads in a other direction and their tail to each other. So and those are protection, and this membrane protects our the cell from the environment. But here the deal. As we know, many different substances and things have to go inside our membrane, cell membrane, through the simple diffusion. It means they're gonna go through the cell membrane, and the researchers have been done, and they find out if your cell membrane is made up from the saturated fats, those membrane is going to be very tough, right? It doesn't have too many spaces between. It's not going to be that liquidy. But if you have in your diet a lot of unsaturated fats, right, like oil, from plants, they all oils from plants, they are unsaturated fats. It means they have a space between those tails, and what's gonna happen, they're gonna improve diffusion in fluidity of your cell membrane. So many different substances can go much easier through the uh, cell membrane, because those tails gonna make those spaces between, and many different substances can go along much easier, like vitamins and different stuff. So that's why people who have um, unsaturated fats and oils in their diet, they have less depression, because the as you can imagine, um, for example, your neurons in your brain have to communicate all the time, and those communication has to be extremely efficient. And if communication has to be efficient, those biolayer of fats have to be able to let different let go and through different things very easily. But if you don't have, if this biolayer is very tough and doesn't have too many spaces between many different things, like uh, neurotransmitters, which is gonna help to go through the cell membrane uh, to help to communicate between neurons, it's gonna be difficult. So that's why if you ha- that's why the if so if neurons in your brain have a cell membrane made from the uh, such unsaturated fats, they gonna, so you have spaces, it's gonna help to it's gonna help different neurotransmitters to the neurotransmitters at molecules which is promote uh, signals between two neurons, so the neurons can communicate, and those neurotransmitters can go very easily through the cell membrane. But if your bilayer is made from the saturated fats, the signals cannot go too well. It's going to be difficult, because all the spaces is going to be taken, right? You have no spaces. You have no space between those tails, and that's why your brain is not doing well. Your memory is not well, because those basically your neurons cannot communicate efficiently, and they could. Uh, and the way to make those molecules from simple to complex chains is exactly the same: dehydration, hydrolysis. But in this case, we're gonna add 3 fatty acids. Uh, so, and that's what I say, king, right? When you have a double bond. So it's exactly the same. You're just gonna add 3 water. You're gonna lose 3 water, or you're gonna add 3 water. But exactly the same concept. So one more time, the protein we're gonna test with a Biuret reagent. Please remember these carbohydrates. We have monosaccharides and polysaccharides with Benedict, and polysaccharides with iodine. And the lipid we're gonna test with brown paper. It's basically you have a brown paper and you add the lipid or oil, and it's gonna stay on that paper. The best example I can give you, when you eat pizza and you have a nice white t-shirt and you have a drop of oil from the cheese, we know it's never gonna it's gonna stay in your t-shirt, right? Because the lipids, they cannot evaporate as water. So this is basically kind of the test they want to give. Let me introduce the very important concept of having different types of controls: negative control, a positive control. So, or experimental group, which is I'm gonna explain more in details in a virtual labs. So today lecture is gonna be a little bit longer. Um, because we are also going to explain the virtual labs. So, but usually this, like when we do in the class, this is one of the most long in class and most complicated. So I just want to take some time to explain things and make it more interesting, just listening and give real examples. Right. So let's just explain experimental group. Why I have to explain this, because the next topic I'm gonna talk is how to do the experiments, and we're gonna use different types of reagents, like so we have different substances like protein, carbohydrates, and different testing reagent, burette reagent, and I just want to explain how it's gonna be. So for example, you have experimental group, you have a protein, and you put some reagent, which is gonna be burette for protein and Benedict for carbohydrates. Please remember this, right? If you have a sample, which is a protein, you're gonna have a positive result. If you don't have a change in color, right, you have a negative result. But then it comes a negative control. So why do we need negative or even positive control? To check validity of your experiments, right? So we have a positive and negative control samples usually validate the experiment. If a positive control sample give negative results, right? Uh, so what is positive control? Positive control, when you have a known sample, you know this is a protein, and you add the burette reagent, right? You add the burette reagent for the protein, but you know you add the protein. So this is going to be your positive control. So you're going to know you're going to obtain positive results. But if you add the unknown and you got reagent and you have nothing, right? You have no change, then you know uh, you don't have a protein in your sample. But why we do this? We want to know if our reagent is actually working fine, right? So we add known sample, which is going to be protein, so just to check how well your reagent is working. So, but if, for example, if you add your unknown, but what if your reagent is not working fine? So you will get the negative results. So right. So this is how you can validate your results, or negative control. So for example, you add water, and you know if you're gonna add water and some reagent is not gonna give you positive control because it's water. It doesn't have proteins, it doesn't have carbohydrates, it's just water. So your reagent is not gonna change in color, so it's gonna stay the same. So negative control and positive control, it's extremely important to validate your experiment. So the next, let me go to the labs and explain a little bit experiments we're gonna do. So test for sugars. So let me go to the student view. So you're gonna read the K concept overview. So basically, so test for sugars is it's a test for simple sugars. We're gonna test for monosaccharide and disaccharide, simple carbohydrates, and you use your Benedict solution, Benedict test for sugars. So I already explained what is glucose, monosaccharide, disaccharide, 2 molecules of glucose, right? Simple sugars, maltose, glucose, oligosaccharide or polysaccharide, starch, glycogen. We already talked about this. So I'm just gonna go straight to simulation. So what you have to do, you have to take and always follow the methods, right? If you do not understand something, go to the methods and follow each step, and then it's gonna get clear, right? So remember, so you have to take your four tubes, right? I'm gonna just skip it, and also you had to add the solution A, B, C, D. So this is a known solution with a known concentration of simple carbohydrates. So the next step, we're gonna add the Benedict reagent to each one, right? Then you have to mix it using different glass and so on. The next, and you just do with the last one. So the next, you turn your water to boil, because the Benedict solution is not gonna react if you're not gonna add some energy, right? And in these and the heat in these terms is going to be the energy to help the Benedict reaction to proceed. And then you take your tubes and put them in the hot boiling water, and you just wait 10 minutes, and wait 10 minutes, and as you see, it starts to change. The color starts to change, right? So what do? You put them back. And now let me explain a little bit what we get there. So what do we have? We have A, B, C, D, four unknowns, right? So what happened? We have A, no change. Then you have B, change to green, C to orange, and the red. So it means all of them have different concentration of a glucose or carbohydrates, different concentration of carbohydrates in each tube, right? The more you have, the more carbohydrates you have in a tube, it goes to the red color. The less, so the B sample has the less amount of carbohydrates, and C has the medium concentration of a of a of a carbohydrates. So please know how the Benedict react change from blue to different types of colors. So know the red is high concentration of sugars, and the orange is less, and the green ones is most less. And if you don't have anything, so why the A didn't change? Because it doesn't have anything, so it doesn't change the color. So, and you go to the next phases. So it asks me to put the observe the color and put to the records, to the lab data. So I'm not gonna do this, I'm just gonna skip it. And you put your tubes to different columns and verify your results. I'm just gonna skip it. Answer your questions, and then you're gonna complete your lab. Don't forget to submit the lab, and the lab is complete. I'm also gonna post the chapter from the lab manual, where you can find how to do experiments, a little bit more information, and this is going to be useful for this experiment, for this class. So the next one, we're gonna do the proteins. So when you're gonna do those virtual labs, suggest you to take notes, how you did the experiments, what did you do, because maybe some of the questions might come up in the midterm, not in the quizzes but in the midterm, and just for your sake to understand how the experiments works. So the next one is going to be for protein. So we already talked about this, how you make a dipeptide, you make a peptide bond through dehydration, and the way to test this is a Biuret reagent. So, and you have the Biuret reagent turns from light blue to pink if a low concentration of protein is present, in dark purple indicates a high concentration. So this statement is not correct. So, and I will explain why. Because you have also you have a pink color if you have not a protein but a peptide. And remember, the peptide is a chain of amino acids which has less than 50 amino acids, right? The peptide, if you have a peptide in your solution, it's also going to turn pink. But if you have a protein, it's always going to turn purple. And you have a low concentration of a protein, is going to give you pink. But if you have a peptide, if you have a high concentration of a peptide, really high concentration of a peptide, it also can give you the purple color. So now it's more complete. And you just start your simulation. You the same. You go to the methods. You read the methods first, right? You read the methods first, and you have to understand what to do step by step, right? In this case, we're gonna have now more interesting lab because we have positive and negative controls here. So you put 4 tubes, right? And now you have pepsin, albumin, and starch. So the pepsin, this is a protein which breaks down as a protein. The albumin is a protein which is going to help transfer the fats in your bloodstream. So albumin can bind to different proteins and move to your different types of cells. And pepsin you can find in your digestive tract. So the pepsin is going to help to digest proteins. So both of them are proteins with different functions. Then you have starch. So what we're gonna do, we're gonna fill out all of them. I'm just gonna skip this step like that. So already put them. So the first one is pepsin. Then you have albumin. Then you have distilled water, and you have starch. So now I want to, and then you put the Benedict reagent, right, to all of them. But so the first one, pepsin. The second one is albumin. The third one is water, and the fourth is a starch. So this experiment has a positive control, right? So the pepsin and albumin is gonna be a positive control. The third one is gonna be a negative control, and then actually third and fourth is gonna be negative controls. And so let me just mix those guys, and we're gonna see the results. You put them to wash, mix it. Right, so you have purple, pink, bluish, and also bluish color. So what does it mean? The pepsin, you have a high concentration of a protein, high concentration of a pepsin protein, and when you have pink, it means you have low concentration of albumin, right? Low concentration. So that's why it turned pink. But the third one and fourth didn't change, right? But we actually, in this experiment, we know what we add, right? When we test for the sugars, we add different types of sugars when we didn't know what we add. But now we add actually known substances, right? So the third one was distilled water. So this was a negative control, right? This was a negative control because you add distilled water. So what is negative control? Negative control when you add the substance, when you add the in a negative control, you didn't have a substance being tested, right? So you actually gonna know this is gonna give you the negative results, right? So you don't have change of color, so these give you the negative results. The same as starch, because we know starch is not going to react with a purate reagent, because pure reagent is reagent which is going to react with only proteins and peptides. So those ones are basically two negative controls, which is going to validate your purate reagent is actually good, um, and it's good to use and test for proteins, because it change for known substances positive control and negative results uh, for things which is it's not supposed to interact. So this is fine, and you go just I'm gonna skip it. You gonna answer the questions and you will complete your lab. So the next one, uh, is gonna be for fat. This is one of the most easiest ones. So only gonna do is just add some unknowns, right? And they're gonna ask you which solution, right? 15 minutes, and it's kind of have to evaporate, and it's gonna leave the marks, right? So you have D, C, B, and A. So which ones? And you have to answer. So there's 5, or you have to put them in the notes. You're not gonna do this. So basically, after 15 minutes come, some substances evaporate. So for example, this one has evaporate, but in A we can see some shadow, which have been left from the substance. So probably it has some oil substance here. Definitely the D1 has an oil. This is the oil. This is look um, exactly the same as you drop when you eat your pizza and you drop that one drop over cheese in your white t-shirt, and you have that spot. This is look exactly the same, and this substance has something, but it doesn't look like oil. It has something to dry on the surface, and this doesn't have much anything, probably water. So, and you just go in and your questions, and choose different options. Milk, water, for example, the D is oil, right? And try to figure out the other ones, and the lab is complete. Very easy. And the last one is test for starch. So go for test for starch. We're gonna use iodine. So, and let's go for, so in this case you have to do exactly the same. You go through the methods. You go to the methods and you follow your methods. Always when you do experiments, go to the methods first, check if you have everything, and then you can pursue the experiment. So in this case, I'm just gonna skip some steps. So you get your 5 tubes, right? Now you have what do you have? You have starch, you have glucose, you have water, and then you have 2 samples tested for starch. How much starch do you have? This is piece of onion, piece of potato, right? I'm just gonna skip the way to do the obtain the juice from the potato. I'm just gonna skip it right here, and then for onion, right? So this is potato, and this is onion, and then you add iodine to your stuff, and oops, um, and you change it. I mean, not change it, mix it. So let's talk about the different. Let's talk about the results we got. about negative controls and positive controls, because this is gonna come up on a quiz for sure. So first of all, you have starch. So this is known ingredient, and this is gonna act as a positive control. So the first tube is gonna be the positive control, because you know this is starch and it's gonna interact with iodine. So we're gonna put iodine, it's gonna turn from yellow, from brown to dark, right? So you know that starch is gonna give you the positive control. So this is the way to test how well your iodine is give you positive control. So this is to check um, the quality of iodine, if it's even can interact with starch. The next one is two negative controls. Actually, the second one is glucose and then distilled water. So both of them, we know the glucose is a simple sugar, so it's not gonna interact with iodine, and also the distilled water. So we know it's not gonna give. So for example, if iodine will give a positive control with the glucose or distilled water, then we should think, oh, something wrong with our iodine is giving the false positive control, but it shouldn't be, right? So that's why it's a two negative control to check how well iodine is working. And we know it's not supposed to work with the monosaccharides, so it's fine, and it's not supposed to work with the water, so it's fine, right? So the third tube it's only have water, so we know iodine is not interacting with water, and it's not interacting with the simple sugars. So we actually can make a conclusion: those two negative controls giving actually right results, which is negative results, and they're right. And the positive control also give you positive results, which is right. So we know our iodine is fine, and it doesn't, and it's interact with the things it's supposed to interact, and it does not interact with the glucose when it's not supposed to, and it doesn't interact with the water and not providing false positive control. Then you have your two samples, potato and onions. So which one you can guess have a more starch? Obviously potato, right? We all know the potato is uh, highly loaded by starch, and the onion is actually very juicy, right? You know the onions are juicy, and they doesn't have things, and they doesn't have a lot of starch, but they still have some. And there, in those results, you can see the tube number 5 doesn't give you that strong results. So right now, this is experimental results. You want to find out the purpose of those two 4 and 5 is to find out how much how much of a starch you have in each sample. So this is actually your experiment, but uh, if you just will do those experiments alone without those two, will you really be sure, like, will be really sure those validate results and you can trust them? No, because how do you know your iodine working fine, right? How you know iodine is not interacting with water from the potato and onions, because potato and onion has some water there. How do you know this? And some in onions can also have a glucose and simple sugars, and potato too. So how do you know iodine is not going to interact them? So that's why you do that experiments, just to make sure the glucose alone, which you can find in a potato and onions, is not going to give you false positive results. The same with the starch. You want to know your iodine is actually working fine and only with starch. So you have just a starch, and you test iodine with a starch. The same one with the distilled water. So you want to know uh, your iodine is not interacting with water from potato and onions. That's why you have to do these positive and negative controls. So please listen this again and try to figure out what is positive and why do we need positive, what is negative and why do we need negative. And if you're gonna get the positive results in a negative control, or if you're gonna get positive control in negative results. And what does that mean? So you have to understand in each case scenario, why do we do this and why we obtain such results and what they mean, right? So you have to be able to understand why glucose didn't give a positive result and why do we use that, right? And you just go next and use lab data, put in notes, and you finish the lab. So what is it? It's going to be, you have to distribute the tubes, verify them, and answer the question. Done, and you finish the lab, right? So the last thing I'm gonna talk, this is I'm gonna post actually the chapter. I'm gonna post the chapter number 3 from the lab manual, and those are the questions I want you to study. I want you to study those questions. I want you to understand them, right? Uh, I'm gonna also post the chapter so you can read the chapter first and be able to find those answers, or you can find them in the internet, Google, wherever. But you have to know those answers because the next class, right? The next class we're gonna have a quiz on those, and the quiz is gonna have some questions based on those questions. So the quiz, I'm gonna actually, I don't know how many questions it's gonna be, but I'm gonna actually take, for example, if starch is present, what color will the iodine turn, right? I'm gonna take that question, maybe a little bit modified, maybe not starch, maybe I'm gonna use sugar, simple sugar, right? But then you have to be able to understand the question. So sometimes I'm gonna do from the positive question, I'm gonna turn it to negative question. But if you understand the question, you're gonna be able to answer. Or sometimes I'm just gonna take the question, not gonna modify it and put in a quiz. So uh, be just make sure you know those. So I'm gonna post this on the Blackboard. All right. So in this point, I'm gonna end the lecture. Thank you very much.